I will provide you the course material also. Please mind it, whatever the presentation we are giving it, it's not mine, it's your also. So we will share everything, those presentation as well as the course material from where we get. Either we give the soft copy of the paper plus some course material we design. So maybe it takes one day. I will take the prints from the outside and give you a small booklets for you. So Professor Zhang is come with this LCMS and mass spectroscopy again in front of you. Please go ahead, sir.
Okay. Thank you very much. Let's continue yesterday afternoon, the mass spectra and application in plant metabolism analysis. This is yesterday. We only go through the sample preparation and several things we should be keeping in mind about the sample preparation. It's very important. At the earlier stage, in second class, initially we designed to tell more details about the sample preparation for LCMS or GCMS or other type of chromatography analysis. But here, I think I thought we can specialize because there's so many different purposes to study different type of metabolite and also different tissue, right? Just as yesterday I said, the seeds very different from leaf, and the leaf also very different from the root. The extraction, even protein or metabolite from the root is much more difficult, I think, because a lot of the water, right? But the leaf, a lot of biomass, more heavily, like water and dry mass ratio. Like water is about 4:1, but the water, but the root is like 9 or 6, and I think about around 8:1. Means 8 grams you only get 1 gram the dry mass. Most of the water. So that makes but it's like a metabolite profiling. Root usually simpler, the metabolite simpler and easier. There's no other polymers like starch or polysaccharide affected. But in seeds, there are a lot of storage protein of carbohydrate like starch or other polymers. Those makes the metabolomics, which we analyze target only below the 1,000 or sometimes 2,000 below, like tritropine, saponin, or sterol. Those molecules are much more complicated than the other small hormone or the flavonoids. Much difficult. So now the LCMS continue, or GCMS continue to expand their methodologies to apply it to other areas. So like GCMS even have limitation. Later I will continue talk about this. Almost every class I will continue talk some LCMS, GCMS. It's not uniquely or not exclusively. I talk LCMS only in LCMS application in the plant metabolomics. No, always the crossover so that you can really in real case, in real situation. Sometimes we cannot limit one machine in one field. No, like GCMS, they also can apply some soluble, as long as they can evaporate it into the gas, they can easily analyze it. Also, there are several libraries available for those electron ionization interface, and also for those under the condition of 70 electron volts, the condition, the library basically constructed on those conditions. So those are very easy to standardize under different conditions with different samples, so people can easily compare in terms of you have stated retention time and molecular weight and other minor parameters. If you put together, you set up the index, very reproducible, very reproducible. That's why I think not only for the sampling the process, but also for those the machine applied or suitable or best applied to reach your research goals. We will talk. You crossover, you mix the manner, not exclusively only talk about the LCMS or only talk about the ECMS. That's actually not the reality. Yesterday we talked about the mass spectrometry. The core part is about the ionization and also how the way they isolate the ion. So now we know the mass spectra is the most important part. Right? HPLC, we know the regular HPLC, we can analyze some known compound in much abundant levels, like flavonoids in many anthocyanins. We can analyze the HPLC. Those are very reproducible. So those for sure we know, but for many other metabolites in minimal levels, micro level, or even nanomolar level, so those will have to use the high sensitivity with the MS. And also for purpose of identification of a new unknown compound. For example, from this plant you find some new compound, the piece you don't know. So sometimes you don't need specifically to isolate this compound. Use the preparative HPLC. You can use a very big column, loaded larger amounts of a sample, then quickly separate, just collect those very high separation efficiency. So you can quickly get the compound enriched fraction, then probably do other. But use the LCMS, you can get very quick preliminary result. The preliminary, you decide, oh, this is what type of compound. Then you can continue. So this is core part of yesterday we talked about the interface, the different ionization ways and mass analysis. This is also core part of Q-TOF or Q-trap or other ways to isolate the ion, already ionized particles or metabolites, already charged ion, how the like Q-TOF use the time of flight in the electric field, it continues accelerates at the time of travel variation. Those particles or metabolites with different charge and also with different molecular weight will be separated. Yeah, will be very easy separated. And finally, all those metabolites, if in the middle they will try to drop, don't have enough speed, you have to use the field to continue charge, give more energy to accelerate the speed. Then continue separate. Eventually, all of them, finally, the signal will hit the collector, the signal collector, ion collector, finally give a signal. Then use the software to transfer those signals into the MZ molecular to the charge, the ratio. According to the whole process, we can actually do the metabolites, like not only the accuracy, because we also have a MS, pattern mass, to analyze the first mass, the broken, the parent, the precursor ion, the second mass, they are the fragmented ion. So basically we can track which which molecule was broken. So those are the very, this is basically later I will continue to emphasize those parts. I think after this course, I hope after this course finished, you basically have a very impressive I will try to continue impress those knowledge concept and the whole idea into your brain. So will be only once. The dance usually you couldn't be effective, but multiple times. This is like human memory. The feature or some determines we have to study again to enhance the memory. So that's why I want to use this way to continue enhance. This yesterday we talked about the several ionization and also the detector, the mass spectra. They have several types of we will continue talk about the first ionization, several different way, and but in GCMS usually we use electro ionization, but chemical ionization also very often applied. I think today course I will also start to after we have a very good understanding or some basic understanding of LCMS or GCMS, mass spectrometry, how they analyze, how they separate those molecules. Then we will step by step get into data analysis, how it gets analyze those ion peak. Then to interpret those data into the real metabolite result. So there are different type of mass analyzer. The previous is the interface, then the mass analyzer, the core drop, and the time of flight. Actually, today I prepared more slides, give you more detail about those. It's the best for metabolite profiling, for really metabolism, metabolism study. This Q drop is the better for. Yeah, they can do the with the internal standard. They can also do the half the quantification, but the quality, do structure determination, a molecular weight, the molecular formula, the prediction is very good, very accurate. You don't need to separate too much, but because the special type of analysis, mass analyzer also coupled with GC, right? So this, and with MS, they also have ion trap, like Q exactly, Orbitrap, used for proteomics analysis and also for metabolite analysis. It's a very pretty advanced analyzer by Thermo Fisher company. So those are the principle we already described, and those ionization is very important. Now applied really in many different ways. So yeah, this again is I come to talk, and this from comparison of the Orbitrap and Q-top or the Q core drop. So we can see the efficiency and the detection, the range of the molecules is very different. They also new methodology development also consider the separation part, because separation part is very essential. Yeah, all the limited top. If you have a more accurate separation or more clear separation, for example, LC, UPLC for liquid, or GC for GC and GC, the two division GC. Later I will talk. So those will make the separation much more efficient. Then later on, the mass spectrometry, mass detection will be more accurate, because simpler is will be accurate. Yeah, more accurate. So this is the two-dimensional liquid chromatography also can achieve a much better result. MS, GC and GC also can get very good results. This is just like 2D division. Yeah, those analysis, like this GCMS only have one capillary column, but if you double those, you will achieve much higher resolution of the isolation or purification. So this some of those work, I think I can send you some PDF of my lab work. So if you read some, if you have questions, yeah, we can, because those work I'm very familiar with, so I can really tell you about my personal experience, and I instruct my students to do each step, not only for making the paper, but also to solve some interesting question in the scientific research field, right? So I think, yeah, later on I have all of the PDF there. This is we use for TVs, the kilowatt analysis. Finally, we connect the possibility of kilowatt loading is thinner or thinner. Those loading amounts could be associated with processed the tea, the flavor. Yeah, because some volatiles degraded during processing, the wax degradation generates different type of molecule than the inside the lipid membrane or different. So there are some non-hydroxy fatty acid, and there are also other substances. Probably during during tea processing, inside the cell lipid won't completely destroyed, but the surface will. So this makes a difference. So we think we connect, this paper we connect the particle wax loading is potentially contribute to the tea flavor formation during the tea processing. There's another we found caffeine. Caffeine actually accumulates in the leaf surface. The caffeine can saturate it into out to the leaf, then probably some way they go to the leaf surface. Because caffeine have anti-insect activity or antimicrobial activity. So probably we think that have an ecological function of the leaf, control the leaf surface microbial or some insects. This kind of defense response. So we will continue talk about the LCMS. This today, actually, we try to focus on the LC and GCMS total. Those tools for the metabolomic analysis of two samples. This like I think yeah, it's a very nice cartoon to measure the whole sample preparation, the tissue and the organ. So later I will continue. It's different mass spectral detection or ionization method and analysis. Later on we will continue. We will step by step close to those are very difficult part, and also kind of the boring, but you close to your data. That's you're close to your progress, right? That's the most important thing. So again, let's I just make a very simple the organization for the sample extraction. Again, I try to emphasize the extraction of a plant sample really depend on the way of extraction depend on your purpose. You want to analyze the polar part or the non-polar part. So solvent selection or this option really depend on your purpose. So I said consideration of your targets, metabolites, or only want to scale all the metabolite you must in your sample. You first, you just want to scan all of the metabolites. This we can do the non-target. We can do all. So those usually, if you use the LCMS, we use 80% of methanol. The alcohol, small molecule alcohol, have a very quick penetration into the tissue. Then enhanced with the sonication, and also under the cold, always under the cold condition. Even sonication, you don't exceed 30 minutes. Or better, put the ice into the water bath to maintain the temperature, not overheat. Makes the sample oxidation. And the sample character, some seeds, leaves, roots, very different because inside you usually analyze the fresh tissue because the dry tissue already many things change. So you usually do fresh tissue. Those fresh tissue contain different amounts of the water, so you have to consider those parts. Those water more or less will affect your extraction because you use 80% of methanol. So if you consider the root, probably you add more methanol. You use 90% methanol. Yeah, a little bit adapted to change. Makes your extraction method adapted to your sample. The flower or just a part of the tissue, or like a sample. The recent year also people do the section laser capture. Just use very thin under microscope, very thin tissues like root tissue. I think a professor in Duke University is a U.S. Academy members. So their group actually they cut the root tips into different sections, different layer of the cell, like on the top, they're called what cells on the root tip. There's some the larger cell or the protection, but the other different layer, they do the single cell transcriptomic. They already understand the science paper understand our different layer of the cell, especially different type of genes. So like oxygen transporter, the only expressed in the bottom layer of the cell, like no matter epidermal cell or cortical cell or other, they always on the one side. The polar localization of PIN1 protein is very important because this is the exporter. They only allow the oxygen from top to the tip, from top to the tip part. So just form oxygen gradients to facilitate the root growth and elongation. So those also achieved by those kind of the sampling. And also, if we analyze phloem or xylem cell, they also have different ways to collect. Sometimes you put if you want to collect this the phloem or xylem, you can cut apart and then put deliquid the cotton and put it inside. I think there have a respiration force to drive the water from soil to the top to the leaf. So you keep one part, the water will continue through the xylem come up. Then you collect the xylem. That's a xylem step already started many years ago, but still we don't know inside is any, besides the amino acid, we know the nitrogen fixation will occur at root, right? Nitrogen fix, the lot of amino acid or other nitrogen carrier transport from respond for transport of nitrogen from the root to the bud tissue, and also from top to the bottom, transported through the phloem, right? It should be PLO, the phloem. The phloem also, I think I have one sample, one PPT to show those kind of very interesting work to understand. And use the transport of the substance root to shoot the crosstalk or communication through the different metabolites, or even those metabolites, some of them act as a signaling molecule to manage the plant, the response of physiological reaction in a whole. So those also very important. I think recent years many interesting papers have been come out. So also maintenance of nature property of metabolites in plant sample. You want to keep the chemical structure not being oxidized or the property not changed by the modification. So this is very important to prevent the sample from oxidation, the further reaction or modification. Just like one example is when we making tea. If we put here, leave them alone, they will continue become black tea will continue to become brown, right? Finally, it oxidize very quick. So those reactions we almost couldn't stop. It's just too much polyphenol in those compounds. So how to prevent those further oxidation? So this is also particularly in some sample, like in tea plants, you extract out the protein sample. You have to add some chemicals or polymer to the extraction buffer to prevent oxidation. Yeah, for those plants that contain high amounts of polyphenol or phenolic compound, you have to add some oxidation, anti-oxidation, or some polymer like PET or PVTP, all those. That's very common. And even we extract RNA from those samples. With the kits, that's fine. The kit already developed contain those compounds. But if you do by yourself, you have to consider those RNA extraction. Similar mechanism for metabolite extraction. Also pre-treatment, the desalting the sample from various culture medium, you have to desalt. Then desalting the culture liquid.
What can we use for desalting?
Yeah, there are PD10 media column.
We can use it.
Yeah, those called I think PD10 column. We can. I think in India also there's the industry makes their own products because PD10 is pretty expensive. From I think from Dubai or from Sigma. Yeah, they prepared those, but very expensive at that time. But later in China, China company just limited those yeah, mimic those products. You know, they produce very cheap the materials to fill the column, have very excellent the desalting effect. So, and also plant extract before loading on the GS. And also another way is to use the TLC to quickly isolate the previous. You can do two division. The first division, you do the use the different solvent ratio solvent development. Then second, the change the solvent. You can do another. Then cut off the single part. Then use the chloroform or methanol to extract the parts. Then apply it on HPLC or GCMS. You can quickly identify this compound. So this is also another way. That's I think some professors already preferred. Our lab also always keep some plates there if we have some structure, chemical structure want to be determined, we have to use those. And also, sometimes when we analyze the flavonoid, we have to hydrolyze with the acid, just for convenient detection. For most of us, makes the complicated metabolite into simplified. That's one very smart way. For example, we isolate the flavonoid. There's a lot of flavonoid in the spinach leaf. Yeah, spinach leaf actually is a lot of methylated flavonoid. Yeah, methylated, and also they have a very tremendous the medical function. Actually, slowly some people realize now some company they try to understand those compounds. Yeah, just visit those compounds. Probably you will find some very interesting finding. Yeah, so for analysis of spinach flavonoid, if you want to find new stuff, yeah, just hydrolyze those flavonoid extract with acid. Then you can get a glycol. Yeah, analyze a glycol, and if someone not not hydrolyzed, probably because those probably because those glucoside is C glucoside. How come? Yeah, bottom actually is how we use the acid hydrolysis to distinguish C glucoside or C glycoside, not only glucose but also the Reynolds or xylene, those kind of sugars. But C glucoside difficult to hydrolyze by the acid. But O glucoside very easy. The most common flavonoid glucoside are O glucoside. The C glucoside have very special chemical property. They have a lot of medical use. Yes, so this just several example how we analyze, how we prepare the sample for the simple, quicker identification of if you have some new compound, interesting compound, you use the regular way to get rid of the other non-interesting and to find some interesting new compound. So to do the further study. So I just list the general way of sample mixture first separated by the L02. Yeah, this general procedure. After LC, then HPLC or UPLC or GC, gas chromatography, capillary column separation. Those separation is through both mobile phase in LCMS. The mobile phase is synthetic. You can combine the different pH or different hydrophobic or hydrophilic those solvents, depending on your mixture. For example, use analyze the carotenoids. Then a lot of chloroform or ester, the plus methanol. And if you analyze alkaloid, have a very strong, the positive charge, also containing the nitrogen. Those, then you have to adjust the pH. You already need to, if you apply the HPLC, you have to adjust the pH to get the very good separation. So this is very important, the first part, the consideration of mobile phase. Of course, gas chromatography, you only can use some, we call the lazy gas, or non-reactive gas, like the helium. The solid phase, also in gas chromatography, is called stationary phase. Is very long. Those stationary phases, because in gas, later on we will measure the gas spectrometry, how to maintain those regularly to wash column with a high temperature. Because temperature high is very essential for the sample evaporation. Yeah, this is very important. If your sample couldn't be evaporated, you couldn't get the analyzed. So this is one of the limitations. But for the LCMS, the concern is about separation, better separation. Because the separation is always limited. That's why some people solve the problem from a mass spectral part. Use the Albi trap, to enhance the different way, to enhance the ion separation after ionization, use chemical ionization. When we extract the lipid, we will show the different chemical ionization. We add the ammonium to let them charged with ammonium, or charged with sodium or potassium. We intentionally add those chemicals to treat those lipids to let them charged. Then later on, we will be easier to get those mass spectra during time flight of during Q-top analysis. So this ionization is we need to consider in how the process can reach the best result, our goal. So generally after those separation, then those analyze the molecules isolated to the MS for detection. There's supported by the mobile phase, the carrier gas in GC and elution buffer, different synthetic elution buffer in LCMS, which continue flow through the LCMS into mass where it evacuates by vacuum system. In GCMS, they have a vacuum system. Later we will talk in the maintenance of our limitation. Our problem happened in GCMS. Many often the vacuum system have leak, cause the contamination. So the sample is first introduced continued by the auto sampling. Right now most of them auto sampling, unless you try to do the preparative HPLC. Preparative HPLC or other the fast fluid it's always for isolation of a large amount of chemical for prepare. Yes,
It's a flash chromatography.
The quick flash you use this to prepare some large amounts of a sample to the biochemical or other is very important. So auto sampling, this really depends on the machine, but most for analytical, the machine we use auto sampling. Enter in together with and also for the GCMS, there are some, most samples are liquid. We extract the liquid, dissolved in the, for GCMS, most often we, for enzyme reaction, we use the hex to extract the substrate and products, then for analyze. And also for those monoterpene reaction, usually we have to, in the chemical reaction, we isolate the enzyme, isolate the gene, express the enzyme in the bacteria, then purify bacteria do the reaction. Usually we put 1 layer of hexane to produce the products will emit from the solution. So put 1 layer of hexane, then after that we extract quickly, load on at least those the solute, right? Solution. But sometimes the GCMS we load in with the gas. This is called the solid phase extraction. Sometimes we use the fiber. This fiber put on the head space of a container inside a plant sample, the dry sample, tea sample, or other plant emits some the fragrance. You put this fiber, they will slowly get saturated, absorb all the fragrance. Then you take the fiber out and put it into, this is swimming GCMS. But another thing also, just to use syringe injects the gas part of the space. You also can detect. But also have different ways to, for the GCMS have some gas state of sample. So mobile phase after separation, then the GCMS or LCMS and GCMS is not as yesterday, so we don't need the baseline resolution. But for the LCMS, usually we need to care about the baseline. Sometimes baseline fly over, so this causes a lot of trouble. So LCMS sometimes easy, but the GCMS is rather simpler. The neutral molecule goes through the heat transfer line into the mass spectra. You know, then in the mass spectrometer, the neutral molecules are first ionized, then the most commonly by the electroionization. This is in GCMS, but in LCMS, most often we use electrospray ionization. And before this, some use those chemical or other type of ionization for large molecules. In electroionization, produced by the filament in the accelerator within 70 electron volts, and knocks the electron out of the molecule. This is molecular mechanism how the molecule will charge. After knock and replace, then produce a molecule that is radical cation. That's a high energy ionization can result in the unstable molecule ion. Yeah, because the change an electron can make the molecule unstable. Then if you have excess energy, it can be broken through the chemical bond, and then broken into the small metabolite fragment. So the broken bonds can lead to the loss of radical or neutral molecule, even larger or smaller. That's usually have to certain chemical. Those broken have a very fixed rules, which part later on we will talk, use one example. And I think later on I will also use flavonoid and analysis, very common. You can see literature, very common. I will take 1 or 2 examples. See, like besides the lipid, those very complicated molecules, and also flavonoids, this kind of very common, it's a very simple molecule. How they have a fixed broken bond, so which you target usually, then can generate the not only the radical but also the neutral molecule. And those molecules rearranged, they can also occur. This results in sometimes very large or smaller. A number of ions, the different mass. So those heavier to bring the molecular ions with fragment ion for very lower masses. This depends on the molecular formula. So, and also for molecular structure of analytes. The structure different and also can be different. So many chemical isomer, we know the isomer have a same structure in the same chemical formula, but different structure. Yeah, so we call it isomer. So those are in some way very difficult to isolate. However, in other way, easier. This is not fully complete, only depend on the machine unit. For example, HPLC, we use the regular HPLC, we use the proper protocol, we can easily isolate EGCT, the major catechins in T plant, in T leaves. We can isolate EGCT and GCT. EGCT and GCT are isomer. Yeah, same molecular weight, but just have the chemical like the head, like the head products, right? The chemicals. One is the inside, another is outside. So those arrange the difference. However, use some mass spectrometer, you cannot separate. You cannot separate. Even not better than the HPLC. We use the HPLC better program. We can easily separate the EGCT and the GCT because those different T plant have a different the proportion of the content in GCT and EGCT. So really not depend on it really depend on the machine. Yeah. Where the bond broken has occurred and which part has remained charged, we always can through the LCMS to get the signal. The next step. What's this? Fix now.
This is mouse.
No. Good. This I will talk later use photos or pictures to demonstrate what's the difference of top and Q-trap over triple George. So the common part is the unit mass instrument, the mass, tadal mass, and the core drop, and also basically this is the core part, is the molecular to the charge ratio. This is the core part. Every time when you see those, you will see that. So because very overweighted to allow only the ion of a certain, this is like a core drop, only like certain MZ ratio molecule have a stable trajectory to through the full port to reach the ion detector. Of course, later on we will detect the core drop Q1, Q2, or Q3. Each have a different purpose of the connection. Sometimes have ion trap and sometimes have accelerator or filtration. Core drop instrument are using the two different modes of operation. Yeah, for lipid analysis, we also use the core drop. Actually, use the triple core drop to reach the goal to analyze so complicate the lipid molecule. And also, For full scan mode, you should have two different mode of operation. For full scan mode, all ions are quiet across the mass range used for identification of unknown compound or method development of qualitative and quantitative analysis for high concentration of analysis. And also the select ion monitoring. This later on will all mention a lot because in analyze some very complex group of metabolites, you have to use this selected ion monitoring mode to analyze. In that way, it's only selected ions that represent the target compound are acquired. Because during the large molecule, the broken down, the only certain, the regularly broken the fragments can be represent the lipid, represent this large molecule, can distinguish this molecule from other molecule in similar group of, for example, phospholipid. The head part is very unique, PC and PE, PI. The only difference, major difference is their hand parts, polar hand parts, like choline or serine. Serine for PI, PS, or inositol for PI, phosphatidyl inositol. So inositol is different. The phosphatidyl serine, this amino acid is different. And choline, phosphatidyl choline. And also phosphatidyl phosphate. The acid is this also polar. They only have a phosphate part. So this will be important. And also use those representative the fragment or signals to be applied. Then this will be useful for the trace analysis and also high sensitivity reach. And we have this, I think, if I don't have I will just quickly go through several times and mention the 2 ion trap. And this is the one of human blood LCMS analysis sample. Yeah, just use the LCMS to analyze the human blood after, the centrifuge. You mix the blood cell to the debris. Then the supercontents come yellow. They call the plasma or plasma part. You log in to LCMS. You can see those clouds for free fatty acid, vitamin E, and then diglycerol or plastosterol ester or triglycerol. That's why after the blood test, they will tell you, okay, you have a high TAG, high glycerol. Then you will pay attention to, okay, I need to take care of, manage my diet, so not eat too much fatty acid or some oil. This is analyze the phloem sap. The phloem sap is from top, from leaf, by synthetic metabolites down to the root to neutralize the root, because the root is very important, right? To plant, they need to manage each part better. So those contain a lot of amino acids. Usually we saw the amino acids is from root to the top, the first one. Yeah, this, okay, yeah. This one. The second metabolite, like doesn't work. But anyway, those metabolites really tell some very meaningful. Probably, if they use a different internal standard, also probably they can isolate some our high sensitivities. Probably can isolate salicylic acid and just monitor those the hormone or signal molecule from the phloem. And this is the use the gas chromatograph to show the hydrocarbon. Yeah, methylated some sugar or the hydrocarbon is like lipid. Yeah, lipid methylated lipid. Just use I think you only use methanol plus sodium hydroxide. Yes, sodium hydroxide to methylate the sample, then can found those different chemicals, different hydrocarbon, the carbon 15, carbon 14, or all those to the carbon 34 from lower to higher. So you can reach, when you reach the carbon 34, the molecule become much larger. That's why basically arrange like with the carbon number. The carbon number increase means the molecule larger, and the charge is very similar. Yeah, so you can get like this. The LCMS. This is another slide to show that I still prefer to show the see that the GCMS is 3D mariner provided the mass spectra that can be used for identify the conformation or to identify the unknown the compound plus the chromatograph can be used for the quantitative and quantitative if you have an internal standard. Yeah, it will be similar with similar property. For example, if we isolate, if we want to measure fatty acid, usually we use the carbon 17 glucoside and the glycerol. Means this glycerol not like a common or natural fatty acid. In the natural triglycerol is usually natural fatty acid is like carbon C12, C14, C16, then C18 and C20. Every time add 2 carbons, right? Every time add 2 carbons to increase the fatty acid, the longest. However, 17 is not common. So we use this 17. Glycerol 3 hydroxy have 17 common fatty acid. So use this synthetic as an internal standard. We do the same way with the extraction and also use the methanol and sodium. Sometimes we use potassium, the sodium hydroxy or hydroxy potassium mixed together. Just like we also also called saponification, right? Yeah, we make the ester. Yeah, make the ester. Yeah, like under those acid and the methyl group added to the acid and remove the one water molecule. That generates ester. The ester even larger molecule, but still it can be evaporated, so it can be analyzed with the GCMS. This is the internal standard needed to be similar to the fatty acid, natural fatty acid. Also for analyze like a terpenoid, usually we use some terpenoid not exist in this plant. Yeah, for example, we use vanillin. Use vanillin is small phenolic compound as the internal standard because this is very easy observed from GCMS spectrum. we can put this together with some phenolic analysis in some compound, in some plants that don't have vanillin. So use this as a, in some other cases we use the purine as the internal standard. It's just common, but only present in purine plant, which is like legume plant produce a very large stalk root, right? Yeah, can be used in medicine plant. So all those you can use different internal standards to do the quantitative analysis. So the three division GCMS is the core drop mass spectrum. You can reach like this figure shows the intensity and also skin number, our retention time, then also the molecular weight to the charge ratio. You also have a very common commercial library with the electron ionization spectrum produced at 70 electron watts. The library can be very easy to use. And also recently, they also have those different chemical ionization, pre-treat the sample, and you can achieve the better result. There's some publication to enhance the GCMS analysis. So very good actually. This is the interface of some, there are many GCMS or LCMS, the producer. So they have different type of the software to make you feel very easier, or sometimes feel unfriendly. Some you feel really friendly, very easy to manipulate. You want to extract the data, or you want to click this speaker and then quickly show up the mass spectra or the MZ ratio, and also give you best match of predict molecule from their own library. So then you can analyze really in detail what's the other the published paper show the similar kind of molecule retention time. It's very close in your materials, the retention time, and you use the same, very similar column, capillary column. The most important is that stationary phase is what chemical property, because those gas carry your sample of substance through those also have a selection. They also can bind to the stationary phase to retard those continue going on. Then eventually, when temperature going down or continue over time, they will come out to the mass spectra. They will dilute out because the carrier gas continues to flow. Then eventually they will pull this power. Then after that, after one sample finished, they will continue use the high temperature quickly clean up the column, capillary column again to make the attached or retarding those chemicals quickly get away. Yeah, just like UPLC or HPLC. After that, we have to use several minutes of use very high charge. If we use the reverse, the freeze HPLC, we normally put the reverse freeze HPLC means that we don't use the charged or highly charged the material to do the column. The material is neutral. Normally use carbon 14, right? Carbon 14, carbon 18, they don't charge. But we use different mobile phase. We use other mobile phase. This way makes adapted to different types of chemical isolation column for the way. But if you use the other in the ammonium or they will make the column property use for specific purpose to isolate. Use the reverse will be so you can. This is another. I think another is this kind of you can give you the fix. You match the something. Not right. Yeah. Yeah. The MS actually is for many usually first the machine I want to buy yes, then consider cheaper and you do many goals. GCMS double GCMS the GCMS 2D regional LC achieve first the separation prevent much better separation and later on MS control the mass the isolation over the determination better because they have a library, right? Library, so this way increase the accuracy, increase the not only because it also can quantitative and so the two division they are developing some software to visualize this kind of colorful visualization of a metabolite. Just recently some software puts the data into the picture. Looks much better, but this is the 3, the GC, then times another 2 GC. This makes the thing even better. Of course, this I think this paper can try to achieve a special purpose. But anyway, there are some common parameters for the liquid stationary phase. For example, their diameter, the capillary diameter is like 0.1 to 0.25. But how much UPLC? UPLC is about 1 or 2, right? 1 or 2. No, it's micrometer, right? Yeah. Similar. It's short, about centimeter. This is one of actually usually when we see our software to analyze, we'll see sometimes shows TIC. The TIC, the concept is the total ion chromatography. So usually you show the TIC in all the metabolites you can analyze shows. So the first you can do the TIC comparison from a different tissue, for example root, stem, and the seeds. You can get the full idea. Okay, under the same identical condition, the different tissue have all those different chromatography of the total ions. This is one way to digest the GCMS. For example, this is for the decay, one of long fatty acid. The decay is the carbon 10 and also just hydrocarbon. Have a 10 carbon and hydrogen 22 hydrocarbon. This straight chain hydrocarbon. So from this mass spectrum, we show this is the molecule, right? Molecule side. Molecule side ion peak is 142. relatively close to it. They 10 carbon, and how much? The 120, right? 120 plus 22. The proton, yeah, it's 140. That's how they show up the MZ ratio. Then we know this compound after electroionization, they will get into use the neutral, get into the mass spectra, then recharge them. Charge them during the flying. There, they should have some part being broken down. Yeah, after ionization, they become stable, unstable, right? They will become fragmented. So first is the two carbon and then 5 hydrogen will reduce. Then give another. Why is the signal very weak? Just because they are neutral molecules, not very high charge. So the signal not very weak. And here signal also very weak. But then you can see the basic way is from actually is you can see the regular 40, 43, 57, 71, and 85, 99, and 113. Those step by step increase 14 MD. Yeah, just because the CH2, yeah, CH2 continue increase. Yeah, initial decrease, they have a CH5, right? CH3, the CH2 because they are in the end, right? Yeah. So those successive overlapping of cleavage of the bond at successive C2H2 unit reflected by the decay mass spectral analyzing, to reflect how we calculate those, how we determine the structure. Yeah, we can understand from different mass ion peak. We know that we can make the prediction of the structure. So this is a kind of math to continue twice mass connect together to covalent the compound that generates the molecule of fragment ion that have a same, but if they have a different structure, even have a similar molecular weight, it could be after ionization, then they can be selected. And the first mass selected is called the precursor ion. The first, because it's a double mass, tedial mass, right? The first mass, the pass by, we call it the precursor ion. The precursor ion will go next, go further, usually achieve the slow and inert gas, the argon. Yes, through this one process. Through this process collision induced deassociation, CID, this is another conception. Then further broken down, produce the ion called the product ion. They continue. This product ion will depend on the structure of the precursor ion. So later we will talk about how they use use the selective ion as the marker of a certain product or certain metabolite. So later we only check if they have this type of ion peak, then we can determine if the standard for the molecule what they stand for is present or absent. The product ion mass spectra for the interference will be different from that of target. I mean, if you have two similar similar molecule compounds, but they have different structure, so they will be charged different. Through the second method, or through the CID, collision-induced dissociation, it can be separated. It can be different, distinguished from each other. This is like using the ion monorating as same mode to do the photo. The first step is isolation precursor ion in the same mode. In the selected ion, the monitoration mode. Then you can see the black and this is green and the circle molecules mixed together. But in second, the collision cell, the fragment precursor, the precursor ion was fragmented. That generates the new green and the blue, the fragments. So the fragment continue through the analytical multiple products ion, and they continue pass through the third MS. They probably further fragmented. If not, that means we're stable. Then in detector, they will collect those same signals. This is a triple photo. The Q1 is used for precursor ion selection. That means that in MS first one, you can analyze. It really depends on you. All of the many molecules and many ion peaks that show up in first part, because in software it will show the first MS is what to give you a. Then you need to scan it. You need to enter into your ion molecule. For example, precursor. For example, your chemical molecule weight is 700. Then you enter 700, it will show up the mass. It will show up the ion peak. But if this ion peak is because 700, there are many compounds with 700, the molecule weight of charge, you have to, according to the retention time, you have to do that. That way, how to compare with the UC internal standard. For example, for those phospholipid, we have the internal standard. We can buy the pure chemicals, phosphatidylcholine or phosphatidyl inositol or PIPCP, PIS, or what are other PA, the phosphatidic acid. We also can purchase most of those. Then we use those standard to do the same way. Then we will see the first part. We get the signal with the same retention time. Then in our sample, we also observe those 700 the molecule with so we can take that. Triple quadruple. That's just the purpose, the whole process. And also the purpose and the procedure. They go through and what's the goal you want to reach. Yeah, just like this. And later on we will have a very good example to show how the triple can be used. Triple quadruple can be used for analyzing larger molecule of that metabolite. This Q3 is for products broken down the fragment ion mass analyzer. And usually the Q3, some signal, very unique fragment can be used to prevent the precursor, means the original metabolite is the mother like molecule. Those also we call the daughter molecule. These are broken down from a certain part of a bond. The whole structure the triple core drop as the most common as the unit mass instrument and therefore more affordable. The core drop also because of the affordability. There are also other multivariation monitoring mode can be used. The Q-top is different. Again, we mentioned Q-top. The Q-top as I mentioned yesterday, most often we do the metabolite, whole metabolite, we use the Q-top, LC Q-top, but also plus the interface we use the ESI, the electrospray ionization. The way of ionization, you make your sample charged. You throw the after sample quickly evaporate, use high speed evaporate, then also be charged in the electric field. Then through the long-term flying through this way, flying according to the this description, time of flight, mass analyze, separate ion waste. After the UPLC or HPLC separation, we will continue to separate those charged ion in the mass spectrum. This is not a vacuum. The gas chromatography, they use the vacuum. Yeah, because gas, but for liquid, no. They already feel some dirty stuff. So based on the time they take to travel down the flight tube to reach the ion detector. So if the energy not enough, you can adjust from your interface, your manipulative part, you can increase it. That's why you can have some flexibility to adjust the proper energy to in charge of continue to accelerate, have a first accelerate and second accelerate electrode to make them continue fly and reach the. So those like a high throughput, just one sample injection, you can get the 100,000 of metabolite. Yeah, so the very high throughput. Throughput, you also you have auto sampling. You prepare the sample later I will mention it. We will talk about again. This one, because of very high sensitivity and also very, very high selectivity, so you cannot use too high concentration. You have to dilute it. You have to dilute it very well. You like a 10-fold or 30-fold dilution, according to our normal extraction. For example, we use you will use 100 milligram of sample, fresh sample, then dissolve in the 80% of methanol. Then we quickly sonication under the cold temperature. Then sometimes we put this overnight, like from 8 o'clock evening. Then next morning we take this in 4 degrees, just left rotation extraction, you fully extract. Then second morning we do the certification. Yeah, use 12,000 table certificate machine, because we only extract 1 mil. We don't need to extract a lot. This is the LC, and that's a very small scale, so very high sensitivity analyzer. Then after certification, we do the memory filter to remove other large particles, because this will destroy too large particles will destroy the injection part, right, to protect the machine. Even in the HPLC, we still do the 0.22 micro molar, the pole. What company usually use for the memory filter? Millipore, right? Yeah, Millipore use the filtration, prepare sample, then we do the injection like a. Normally use HPLC, we use the injection 5 microliter or 10 microliter or sometimes. But in this high sensitivity machine, you have to 0.5 micrometer. That's enough. The very sensitivity very high, and also give you don't need to worry about the injection to a small amount. You couldn't get the signal. No. This with the same kinetic energy. Those with low masses have a high velocity, high speed, and therefore arrive first. Whereas those with high masses have a low velocity will arrive later. So this can separate according to molecular weight. And top MS instrument can also arrange the mass solution and the acquisition rate very fast, the top one, and also with the acquisition rate up to 100 spectra second rate. And also the masses, you can do selection, like a molecular weight selection. You can select from a small molecule to like 200 above, or like 200, 400, because if you interested in molecule with this molecular weight, you can focus on this. But if you want to get the larger molecule like saponin, okay, you can take the range from 500 to 150. Around this molecular range usually cover most the saponin. Yeah, this recently saponin study very cool. Several science papers recently come out by Stanford University and also Joint Center. Their paper for practicing. The campaigning potential of OB trap with the mass spectrometer is also a high resolution platform, or even called ultra high resolution. We call it ultra high because this machine just uses a different way to give from the total ion chromatography, TIC, the chromatography. You see very dirty, so many different, no separation, not like normally if you run HPLC very often, you will see, oh, this total ion, the chromatography is too dirty, too many, no separation. But actually, use this special machine, you don't look at those total ion chromatography. You want to check you heat anywhere, they will give the mass spectrum. This is really the nice product. So how they work to get this much high resolution? Just because they use some not only let's just read through, not only for high-end proteomics application. That machine usually can do the two types of service. One type you do proteomic study. Yeah, if you have a sample, I want to do the proteomic study, you can use this machine. Yeah, just the big box. Yeah, from outside you couldn't see. Now the machine just not like an old machine. From outside you can OB trap so long. The corridor of electric field. But for this machine, we covered just a big box, and you apply the sample, and you get the proteomic data. Then you analyze the proteomic, of course, you need to do some pre-digestion, right? Use the tyrosine or some protein. Yeah, protein is the enzyme to digest your whole protein in different fragments. Because you have reference gene or sometimes you can set up your library so you can match different fragments. They were just like sequences. They will know the peptide, the molecule, and also they basically know the separation, the sequence of the amino acids of small peptide. So that's very, very nice the machine. So you can do the protein fingerprint study. So you merge the protein according to this. For example, if you want to study the metabolite and protein interaction, of what kind of the protein bind to this lipid or bind to this compound. So you can use the compound to probably make into resin, line to resin, then pass through your plant. Maybe some protein bind to this, then you look at protein to this analysis to find the really, the truly interacted the protein or metabolite. Or sometimes you put the protein there, then you can use the protein as a way to trap the other molecule to find that all the molecules can bind to the protein. Doesn't matter is in the physiological condition to do or probably do. Yeah, sometimes we just, because we extract the plant, then do the in vitro binding, doesn't mean they really bind in the cell. But this at least indicates some, just one way of advanced study this. So we haven't published the result yet. Very interesting. The machine can be used for that kind of purpose. But also, this is one of the principle to analyze use the spin, this ion trap can orbit just like in our planet, the Earth, as a planet, planet turn around with around the sun. Yeah, in polar, in the galactic system. So this also just that's the molecule with different charge, electron charge, and with different molecular weight to turn around in different areas and continue different routine. Of course, this we have to do the selection, do filtration. So from the sample, then through the ultra high field of orbitrap mass analyzer, finally we also coupled with the photo mass filter to get the C-trapped molecule. Finally, we get the ion trap mass scanning, gets the data. This is another newly developed. I just read the paper. Very good isolation for the peptide or this the nanoflow, the liquid chromatography. Yes, this different type of LCMS, MS. The pattern mass also can get very good isolation of digested total protein to the proteomic study. I think this is the limits. The GCMS suffers from two major limitations. That's we have to even we like GCMS, but still we have a limit. Not all the molecular or not all the metabolites can be evaporated. Evaporation have usually analyzed for the volatile. So the more stable the compound amenable for the analysis cannot do that. But still, you can do the other derivatization. Yeah, we know they cannot do. But however, if we find the proper way to modify those compounds, for example, Q10, they don't evaporate. But after we do some derivatization, use two chemicals from a sigma. After treatment, you can do very easy isolation through the GCMS. And this method has been used in almost all the Q-tubular wax study. but they don't choose to use LCMS to study the particular wax, just because this very specific can identify all the particular waxes. So that's why we still try to overcome this limited. For some time, really we couldn't be evaporated. No, probably you just haven't find the proper method to methylation or to other. However, anyway, those new methods still under this development. The second, electron ionization mass spectra suffer from frequent absence or weakness of the molecular ion. Yeah, that's also true. That for certain limited compound, the electron ionization have their limits. But if we can change the ionization method in the GCMS, yeah, there are some reports already started, trying to improve it. And those absence of the weak molecular ion result in the reduced lower the confidence level in sample identification by library. Yeah, because the ion peak not enough, so you couldn't find another a strong signal to match the library. So because of those two limitations, the use of GCMS is growing. Yeah, as one of the alternative. So particularly coupled with other different type of mass spectrum, just as mentioned above, like orbital trap or triple trap or other two-top. A problem is when we run GCMS, sometimes like this maintenance, probably we can see some leak into the system and system contamination. Yeah, this is very common when you see this come out the very noise signal and or other unknown stuff. You definitely think your system need to clean up. But the capillary column have lifespan. You have to replace also for the injection in lab parts. Yeah, those need high temperature or sometimes the contaminator or their temperature can go in, you just need to replace the very good strong vacuum is required for reduced background interference and have molecule reaction for good the sensitivity to increase the lifetime of the component, reduce the maintenance and avoid any electric discharge such as high voltage. Yeah, just the small leak can increase the background where the large leak impact the vacuum. Yeah, if large leak, the machine just shut down. High column flow increase the number of gas molecules entering into the mass spectra. Such a system cannot evacuate then fast enough to establish the good vacuum. So the sensitivity reduces. Therefore, removing the method to the GCMS just can be done. The troubleshooting smaller internal diameter column should be used to increase the separation and also can reduce the column flow rate, so not enter into the mass spectrometer too fast and to cause the crowd the molecule on metabolite identification. Maximum flow rate for the high sensitivity. Usually this have negative correlation. If you have too too high the flow rate, we will reduce the sensitivity just because the crowding. Yeah. It depends on the vacuum vacuum system. Also, yeah, vacuum system is very important for the mass, for the GC and mass spectra, for liquid method. The sensitive, the mass spectra requires the regular maintenance of the clean the iron salts and replace the oil in vacuum tube to make sure the vacuum is strong enough to keep the degree of vacuum. Optimizing the inlet temperature. Yeah, this inlet temperature is sample, where sample will inject and then carried by carrier gas mobile, and the mobile fixed in CMS. So those also need regular replacing the GC inlet in turn not only for the contamination but also for damage to other stationary phase. The inlet part is not controlled, not good enough. We will destroy the whole the capillary column. Yeah, just I don't know. In Chinese, they are like 4,000 plus a couple thousand. It depends on the column. Yeah, this is the when during the quantitative analysis or qualitative analysis determines structure or determines the metabolite formula. One of the internal standard or use the other like heavy metal. This is like E6, you know, like heavy water, right? Yeah, you use those. Also, you know, just the hydrogen, the different, the D6. But then, anyway, the acetone of the D6, so you can see the acetone D6 have different molecular weight. Is too heavy. I think we should call it heavy water. Yeah. So the molecular increase 6. Common hydrogen only 1, right? But this is 6. But use this as internal standard except the isotope. Isotope you can do it. You particularly for stable isotope. For the for the LCMS or GCMS, very common. Yeah. So this is just a comparison how we identify the real molecules or do the internal standard. The isotope, yes, the isotope standard or stable isotope standard is very important for the hormone analysis. For hormone analysis, almost people only accept the high level of methodology paper, only except using the isotope labeled chemicals as a standard. You can clearly see that our jasmonate with the carbon 14 or carbon 13, no, carbon 14, the carbon 13 or carbon 12 is different. Yeah, every part is different. So people can see, okay, you use this to identify the retention time and very closely accompanied to the natural jasmonate compared with the carbon 13 stable isotope, isotope labeled the jasmonate as standard. So this we can make sure the Q-top MS is established hormone analysis is reliable. This is yesterday I talked about. I still did not put the lipid extraction procedure, but I think this website you can click that together. This lady is a technician. She another professor. Yeah, she did a lot of the work just to help analyze from all over the world. we China we have sent a lot of those samples to her to analyze. Also European and also the I think other parts of United States send a sample. So you can actually done research center. What they use for this also is the quadruple, triple quadruple. The pattern mass spectrometer with a collision cell, the quadruple, middle side Q2 is a collision cell, called the CID. The chamber where the fragmentation occurs. This is very important. That's why, because the lipid molecule is too large. Between two mass Q1 to Q3, between that region. Then the brand, but they also have other like Q-trap or ABI, the LCMS, the one street, but some of them. So I just give you the basic chemical structure of this molecule. So later on, you will understand when we analyze the determined structure or established method, how those broken down happened. For example, normal lipid, the polar lipid, and the non-lipid, the hydrophobic, non-polar kilo. I also have a polar head. The polar head, the hydrophobic usually is several compounds, for example, PE, ethylamine. Yeah, ethylamine is the polar head, and also the choline is PC. So the mass broken side can be here, and also can be here. To remove this is PE and ethylamine. You can go from both sides. So that can generate the different size of the molecule. So, and also this molecule, this group of molecule, the very one and 2 and some 3. The most often photosynthesis plus in D. Later on I will show that. We know this the broken side. This okay, so the molecule is 1831 and S2DG sulfur containing glycol lipid. You know, there are this glycol molecule, then one hydrogen the sulfur, and another 2 is fatty acid chain. So if you broken down in different parts, you will give each part I give the molecule. But the total part is like 832. When we calculate
So, thank you, Professor Jain. This was a really very good presentation. Getting from the all of you know now a little bit about the spectroscopy that we are using with the LCMS. So now we will take a drawing photographs just downstairs, and then you have now there is no mesh for the lens situation. You will go take the lens and then come again together at the 210 shortly, because we have to finish it at the 410. Then has to go Sirsa, and second there is another event here at the 420. Some other departments have 20-30 minutes to faculty interaction. So we would like to cover those 2 hours, 210 to 410 shortly, and in that